Welcome to another episode of Stridulation Station, brought to you by the Entomological Society of America. This is a show where we support, empower, and connect entomologists by sharing practical career guidance, professional development insights, and community-focused conversations from experts within the field. We explore the many facets of entomology from career paths, insect science advocacy, emerging technologies, and even leadership opportunities to help listeners grow as both scientists and professionals. I'm Danielle Tempesta, the meetings manager for ESA, and I'll be your host. Live attendance to these conversations is an ESA members only benefit. So you can join ESA to tune in to the upcoming conversations. You can engage with the panelists and get your questions answered. The recorded conversations are available free in both video and podcast format about a week or two after the original discussion. So today we're diving into the world of insect science with Dr. Beatrice Dingha and Dr. Tolu Morawo, where we'll talk about reputability, research, and resilience. This conversation is hosted by the International Association of Black Entomologists, so Tara Kate Jones helps moderate the discussion. Our panelists share photos from their academic journeys and career histories. So if you want to see the photos during this discussion, you can watch the video of the episode on YouTube or our ESA Learning Center. Let's get started.
Thanks, Danielle. Well, to all our attendees, thank you for attending. This is a Black History Month celebration. We have our members, which will be amplifying their voice on reputability, research, and resilience. Before I get started, I would like to thank the organizer from IABE, myself, Dr. Tolulope Agonbeade, the Vice President, the Secretary, Dr. Olufemi Ajayi, and our Treasurer, Dr. Blessing Adekemoya. Now I will introduce our panelists, which are members of IABE. Our first panelist is Dr. Tolu Morawo. He's an entomologist and a major at the U.S. Army and Navy as a civilian. Our next panelist is Dr. Beatrice Dingha. She's an entomologist and a professor at North Carolina A&T State University. Both panelists will give a 15-minute presentations. You can post your questions in the chat, and after the presentations, we will have a Q&A where I will pose those questions to the panelists, so you will get an opportunity to hear from them on your questions. Now we'll ask Dr. Morawo to share.
Again, thank you, Tara K, for that introduction. Saves me some time on this slide. My name is Tolu Morawo. I work for the U.S. Army Reserve and also the U.S. Navy. For my part, I'm going to be talking about the intersection of entomology and national security. So first and foremost, I have to get this out of the way. Just a disclaimer, the views expressed in this presentation are those of the author, which is myself, and do not necessarily reflect the official policy or position of the Department of the Army, the Department of the Navy, the Department of Defense, or the U.S. government. So this is a presentation I'm making in a personal capacity. Okay, so a lot of questions that I get is, how does an entomologist fit into the military system? So generally speaking, if you, I'm going to use the Army as an example. If you're in the Army, you have two different type of job codes. You are either in combat arms, that is, you are a warfighter, or you are combat support, that means you're supporting the warfighter. So one of the combat support that we have is medical, and that one will be legal, and then we have administration and the likes. But I fall on the medical. And those who administer that is the Army health system. Under the Army health system, we have 10 medical functions. Some of those ones you will probably recognize, medical evacuations, logistics, dental, but one of them is operational public health, also called preventive medicine. It is under this function that you will find entomologists. They're often embedded in preventive medicine detachments, which are usually at the second role out of the 4 roles of care that we have in the Army. If you look at this front line here, and then you look at the role 1, which is usually the medic. Just next to it, close to the field, is the role 2, which is where you find the preventive medicine or the entomologist. So that kind of gives you an overview of how an entomologist fits into the military system. Now, personally, I refer to myself as a dual capacity DoD entomologist, and that is because sometimes I am the one wearing the civilian attire while other people are wearing uniform when I'm working with the Navy. And sometimes I'm the one wearing the uniform when I'm working with the Army Reserve. So those two things are happening at the same time, and it is a balancing act for me, how I allocate my time between one and the other, but it's something that I've really come to enjoy in my career, as both jobs really complement each other. So now to the real topic, the intersection of entomology and national security. How did these two things come together? I'm going to talk about three main areas of emphasis. One is the vector-borne disease prevention. Another one is property damage prevention, and another one is food security. With vector-borne disease prevention, you think about all the things that can transmit diseases. Think about mosquitoes, ticks, um, kissing bugs, think about, different things. And it is entomologists that are responsible for making sure that there's enough surveillance in areas where service members are going to be doing training or doing their missions, and also making sure that they are taking the proper precaution. And if there are threats in the area in terms of, after going through all the diagnostics, and we're able to make sure that we mitigate those risks. Another one is property damage. Think about something like termites, for example. If we have wooden structures in training areas, they can cause a lot of the damage, which directly translate to, waste of a lot of funds, something that we don't really like to do. Not only that, it also disrupts missions, especially if it's an important structure. And then the third area is food security. There is really no national security without food security. Think about, I know a lot of us here maybe deal with field crop pests, but also think about stored product pests, infesting food. And besides that, we also have livestock pests. A good example is going to be the New World screwworm that I'm going to talk about later on in the presentation. Okay, so let's start with the vector-borne disease prevention. A case study that I'm going to use for one of the research that I did was ticks. All right, we know that ticks transmit a whole lot of different diseases, ranging from anaplasmosis, some of the less known ones, to some of the really popular ones like Lyme disease and Rocky Mountain spotted fever. But our service members are usually always prone to coming in contact with ticks as they train in the field, grassy areas, and sometimes even in recreational areas like a golf course, for example. So what we did was a study that was funded by DoD through the GUIDES program, and we had a couple of years. I was a PI on this particular work, that we did tick surveillance across military installations in the Pacific coast of the US and also in Alaska. In the Pacific coast, including California, we use primarily the tick drag method, as you can see right here with one of my old colleagues using the tick drag to collect the ticks. It's kind of like a passive sampling method. And then, but that did not work for us in Alaska, as we figured out in a hard way. But the following year we had to do what we call live trappings using the shaman trap, and we would usually have voles and shrews come into those traps, and then we'll take them out, pick off the ticks from them, and then release them. And that was how we collected ticks in Alaska. Right here, if you look at on the bottom left side, at least left side for me, you can see a chart showing the results. One thing that would jump out to you is 2021. Um, we just had just massive abundance of ticks in a single day, 600 ticks in one single location, which really skewed that data. But I'm showing it here so that you can see that is real life. That's what happens in the field, and this is why we do what we do. Most of what we got were really exotic species and Damascenta species, all of which can transmit diseases. If you look on the right side, and then we went ahead and tested those ticks for pathogens. And then you can see just a bunch of different types of pathogens that are present. Pay some attention to Rickettsia rickettsii, which causes the Rocky Mountain spotted fever. Overall, we had the positivity rate of about 32%. Typically, what we do with these findings is we analyze the result and then we kind of analyze the risk that the service members are in. And then we advise the commanders of the different installations or the training areas as to how they can better protect their troops. This is the whole point of what we do. Next up that I'm going to talk about is also another research that I did with kissing bugs. They're also called triatomines. For those who have worked with this or coneheads, they cause the Chagas disease. They carry the pathogen Trypanosoma cruzi. In the US, the actual range of the cases are not well known, but it could be up to 1 million in the US. The reason is because a lot of people don't know that they have Chagas that have been infected because some people never just show symptoms, and for years and years it may not show up. But when it does show up, can cause very serious complications, including heart diseases. You can have serious acute and serious chronic symptoms, and a lot of people just don't know that it's going on. But for our service members, they are also prone to coming in contact with kissing bugs, especially as sometimes they have to sleep in the field for some of their trainings. What we did was we started collecting the kissing bugs along the U.S. southern border. We had sites in Oceanside, California, and also in Yuma, Arizona. We will usually go and deconstruct wood rat nests because the kissing bugs are usually associated with the wood rat nest. What we found was we got a lot of Triatoma rubida and also Paratriatoma acuta. These two have been known, and some of the samples we had also carried the Trypanosoma cruzi. Again, just like the previous one, we analyzed the risk and then we advise the troops and the commanders about what to do best to prevent this from happening to the troops. And then now I'm going to switch focus to food security. I'm going to use a case study with the New World screwworm. This is a very devastating pest. The larvae of this fly, they feed on live tissues as opposed to most other fly species where their larvae will grow and develop in decaying organic matter. Specifically very devastating to the cattle industry. I got to be involved in this when I deployed to Honduras last year, and I was the only entomologist in the region. I was called into this to coordinate with the Honduran Ministry of Health as they were preparing their sterile insect technique, and also with the airline contractors as they were helping to disperse the sterile male flies. So this is an old enemy that was, I would say, in military terms, that was already conquered in the US decades ago, but it's making its comeback now from Central America, and now it is in Mexico, and it is actually very close to getting to the US. So because of that, some of the border states have just recently declared a disaster declaration for the New World screwworm infestation, and they are really monitoring that. All right. So still talking about food security, I'm going to take a little step back now to talk about my own graduate program before I even joined the military at all, and some of what I did in plant insect interaction research, which has implications for crop protection. So pretty much for my graduate studies and even for my postdoc, and then going on to the brief time that I spent as a faculty at University of Florida, I was really more of a chemical ecologist in my mind, but insect behavior is really my passion. I was looking at what odor cues generated from the interaction between herbivore and plants, and how the blend of those odor cues can be used by natural enemies such as parasitoids to locate their host. That was really the main thing. But through that, and then going forward, I was able to use some of that knowledge and just the behavior of the natural enemies to see how we can enhance the use of biological control agents to better our programs. While I was at Auburn University, I used the parasitoids, micropris, crossopis, and Cotysia marginiventris. Some of my other model system was using cotton and soybean, and I also used a lot of the noctuid moths, especially the Eleonthes virescens, which is the tobacco bud worm. At University of Florida, I briefly worked on the Asian citrus psyllid and two parasitoid species, and I was just looking at the chemical ecology aspect of how we can use that to enhance the effectiveness or efficacy of the parasitoids. Now, through it all, I would say this, that something that I've learned through my own young career so far is that a resilient mentality is something that you have to go through all of this. But it's not only that, it's also having a supportive community. The combination of those two things, which one you can help, one you may not really be able to help, but sometimes you may be able to choose your coworkers, your mentors. But what is most important is that you surround yourself with a supportive community, and that will bring career success. If you look at the photo that I have here, it's a photo of my Dr. Faramaru's lab, which was he was my graduate advisor. And here you can also see some of our IABE members, including Dr. Dingha, who's also on the panel today. That's especially why I chose that particular photo. So it's good to see everyone's face. But this was my foundation. This were the people that kind of shaped my own research career, my colleagues, my mentors that work with me. So it's very important to have that supportive community around you. Picture here shows my old colleagues at NetMu5 Navy, working with them in the field. And then the last photo here is where I currently work with NAFAC Atlantic in Virginia, where I work as an applied biologist and also a pest management consultant for the U.S. Navy. But at this point, I would like to share, a quick story, which kind of corresponds with this photo here on this graduate school magazine, of my own personal story. In 2011, when I first started my master's program, I started working with parasitoids and looking at their insect behavior in the olfactometer, and it was just not working for me. I was using a traditional kind of horizontal olfactometer, which was kind of the commercially available one. Everybody used that, but the insects were not behaving; they were just not moving. So, but because that was kind of like the known standard, it took a while for me to even, convince myself that maybe I should change the design. But I finally came to that realization with a lot of encouragement from my advisor, and I decided to do a redesign, kind of modify that design with a new olfactometer, which had a longer walking stem, as you can see from this photo. And then it had this angular arms, which kind of increased the aerodynamics of airflow. The result is that it became more efficient and the insects were behaving. But it's just how I got there. It just took a lot of being resilient and just sticking with it, because there were days and days of just being in that olfactometer room, just doing it and nothing is working. But I just kept on going with the support of the people around me, which is the two components that I'm kind of emphasizing here. So that was something that I encountered early on in my career, and that experience kind of shaped me even until now. So I think kind of ran out of time, but if you would really like to read more about my stories, you can scan this QR codes. I know this is being recorded, so you can go back to it, scan this QR codes, and you can read more about my bug stories. I have a lot of people to thank. Here's a list from universities that I've worked at, USDA funding organizations, the people who helped me with my sequencing data, my colleagues at the Army Public Health Center at San Diego State University, um, places that I've worked, and also funding agencies within the DoD. That's it for me. Thank you.
Thank you, Dr. Morawo, for a very insightful presentation showing your research and your reputation and resilience. Now we will pass on to Dr. Dingha.
Good afternoon, everyone, and thank you for the introduction and the opportunity to participate in this important conversation. Today, I will speak on advancing excellence and resilience in Black entomology, from research and discovery to impact. My remarks are drawn from my research experience, mentorship work, and engagement with agricultural systems that directly affect food security and livelihood. First, here is my story of how I got to where I am today. I'm married to Louis Jakai, an entomologist, and we have 2 sons named Leonard and Stanley. My story is a powerful example of resilience expressed through persistence, adaptability, and unwavering commitment to education and excellence. Raised in Cameroon and educated at a prestigious girls' secondary school, I excel academically within an English-speaking system. However, at the time, university education was primarily in French. This created a language barrier that threatened to end my academic journey before I even started. Resilience emerged when I felt the system say no. With determination, I said, continue. Resilience first revealed itself with my willingness to move beyond national borders rather than surrender my ambitions to further my education. Determined to continue my education in English, I applied to universities in Nigeria, leaving home to study in a country where I knew no one and had no support structure. A decision that required courage, self-reliance, and faith in my own capability to succeed under uncertainty. When my first choice program, biochemistry, was unavailable, I demonstrated resilience through adaptability. I accepted admissions into the Department of Parasitology and Entomology, a discipline I had not initially envisioned for myself. Instead of viewing this redirection as a setback, I committed fully, transforming an unexpected opportunity into the foundation of my scientific career. My resilience was tested repeatedly during my undergraduate years as university strikes disrupted academic calendars and forced me to return to Cameroon multiple times. Each interruption brought uncertainty, financial strain, and emotional fatigue. Yet, each time I returned, refusing to allow instability to define the limits of my future. Resilience meant refusing to quit, even when the systems repeatedly stalled. Resilience carried me forward to Ghana, where I earned my MPhil, and later across the Atlantic to the United States to pursue my PhD at Auburn University. I later joined North Carolina A&T State University in 2010, and I have been here since. There have been challenges within academia, as they are everywhere. I never allow these obstacles to define my limits. Through consistent hard work, resilience, sustained productivity, I continue to advance and make meaningful impact. Resilience is not the absence of obstacles. It is the refusal to let obstacles define the end of the story. Excellence in Black entomology is achieved at the intersection of reputability, research, and resilience. And here I term it the 3 R's. And the 3 R's are interconnected, as you can see from the screen here, where reputability establishes trust, research produces evidence and impact, and resilience sustains scientists in inequitable systems. Together, these 3 R's allow Black entomologists to survive, lead, innovate, and transform entomology. So if excellence in Black entomology is achieved at the intersection of these 3 R's, so what are the barriers that have limited the full advancement of Black entomologists, and why should our voices matter? There are several reasons, but I'm going to dwell on a few. First, Florida A&M University is the only HBCU land-grant university offering BS and MS degrees in entomology. Second, limited entomology departments, Merging closely related departments reduces entomology faculty and programs. Thirdly, unequal access to research resources, lower funding rates, or smaller award sizes. Therefore, our voices matter because we bring in diverse perspectives, which enhances research, improves problem solving, and strengthens scientific inquiry. The future of Black entomologists depends on inclusive leadership, a model that intentionally creates opportunities for Black scholars, interdisciplinary research, which would integrate entomology with multiple disciplines and institutions, and more important, community engagement, which aligns scientific excellence with community relevance to serve the people as much as it advances the science that we do. Therefore, moving forward, we must continue to publish to ensure Black excellence in entomology research is documented and cited, mentor intentionally to remove barriers that instill excellence. Systematic challenges may exist, but reputability opens doors, fuels scientific discovery, and magnifies impact. Reputability enables access to funding. Over the years, I've secured over 66 million in grants as PI and co-PI from different funding agencies. By establishing reputability, we gain the resources to conduct meaningful research and provide mentorship that shapes future scientific leaders. Building a strong reputation creates opportunities to mentor students. As you can see on this slide, over the years, I've mentored several undergraduate and graduate students. I've taken my students to meetings. Some of them have won awards. I take them to the farm to train them. And right here is, we usually have a summer program for high school students. So we have summer students come over for internship programs and we do train them. Building a strong reputation creates opportunities for outreach. On this slide, you can see below where the IPM program at North Carolina A&T State University, we celebrated World Bee Day on our campus. And to my left, I'm demonstrating the use of cowpea as an intercrop to increase crop yield during the small farm field day at our university. And to my right, far up, we go and meet our collaborating farmers and engage with them to ensure that the research that we do also has some impact on those farmers. And to my left, to my right below, I'm here engaging inner city summer camp participants at the research plot here at A&T to introduce them to agricultural research and provide them with hands-on exposure to farm life. Reputability advances research excellence, but reputability itself is earned. It is built through persistence in the face of challenges, adherence to integrity in all scientific work, and sustained effort over time. I'll use this as an example to show that reputability is built over time and not instantly. As a graduate student, these were my publications. And the past 5 years to my right is my publication here at A&T. However, reputability goes beyond publication. It involves community engagement and outreach. So what is the take-home message here? The take-home message is that reputability is the bedrock on which scientific credibility is built. It is our most valuable professional asset. So I encourage us to protect it. Research that drives impact is very important because it creates knowledge that changes systems. It strengthens communities and advances sustainable solutions. In the next slides, I'll be talking about some of the research that we have done here at A&T, the impact from this research, and the publication of some of the work that we have done. So we did some work on the brown marmorated stingbok, which is an invasive species first reported in the US in the mid-1990s, spreading and causing economic damage to several vegetable crops and field crops. Beyond agriculture, it is a significant household nuisance in the fall, invading homes and structures. In one of our studies, reliable rearing methods were developed, which are essential for conducting controlled experiments and advancing pest management research. The second study evaluated how brown marmorated stingbok interacts with different colored bell peppers, providing insight into host preference and potential crop vulnerability. Findings were not only published in peer-reviewed journals, but they were also highlighted by Entomology Today. You can see to my far right-hand corner, and many other outlet sources. This recognition reflects the broader relevance of the research, connecting laboratory science to growers' concern and real-world agricultural decision-making. We also did some work on pollinators on cowpea and intercrop. Pollinators we know contribute over 24 billion to the US economy each year and are responsible for producing over 30% of all food crops. However, their abundance and diversity are on the decline. So the work that we do, or shown here, addresses real challenges in agricultural systems, particularly pollinator decline and the potential risk to the production of pollinator-dependent crops. What's important is not only that this research was published, but that the findings were recognized and highlighted by the funding agency, as you can see to my right, demonstrating relevance, credibility, and broader significance. Industrial hemp came into the picture. The US Farm Bill Act of 2018 legalized the production of industrial hemp and allowed for it to be treated like any other agricultural commodity. So we engaged in some research. In one of the studies, we examined knowledge, perception, and interest in the industrial hemp among North Carolina organic farmers, providing insight into adoption barriers, educational needs, and decision-making at the farm level. In another study, we evaluated the chemical composition of industrial hemp pollen and how bees respond to it, directly linking crop production and pollinator health. The fact that these findings were highlighted by Business North Carolina, to my far right here, underscores their broader relevance. It is a signal that this research extends beyond academia and contributes to conversations about agriculture, sustainability, and economic development in the state. We moved on with our industrial hemp because from previous work, we found out that hemp produces pollen, it attracts pollinators, but requires nitrogen. Also from previous work, we found out that cowpea attracts pollinators and fixes atmospheric nitrogen. So we did intercrop hemp, cowpea, and pollinator-dependent crops. By intercropping hemp and cowpea, we enhanced pollinator activity within the system, which translated into increased yields of pollinator-dependent crop. In this case, it was squash. This work highlights how strategic crop diversification can support pollinators while simultaneously improving agricultural outcomes. The findings were highlighted by North Carolina A&T State University's research magazine, underscoring the broader relevance of this work and its value to growers, researchers, and extension audiences. I would like to acknowledge my collaborators, students, funding agencies, and institutional partners who have supported my work. I also thank the leadership and members of IABE for fostering a community that values excellence, representation, and mutual support. Thank you very much.
All right, thank you so much to our two panelists. Now we will get into the Q&A sections. If you have questions for our panelists, please post them through the chat, but I will start. There's a question from Blessing Adekemoya. It is to Dr. Morawo. Thank you, Dr. Morawo, for giving us some insight into your research in the military. Just curious, is your research or field studies strictly determined by the proximity to military bases or activities?
All right, thank you, Blessing, for your question. I was just putting it in the chat. I said, for the most part, my field studies are conducted on military installations. But not only that, sometimes it's places that are close to military installations, kind of the surroundings, or sometimes it's the training areas, or even the actual mission fields, maybe outside the country. The reason why it seems as if we're kind of restricted to those areas is because our target is to protect our troops. So wherever they walk, wherever they train, wherever they live, that is where field work is. So that's kind of how that's determined. But one good thing about it is that the community also benefits from some of the things that we do just as much as we benefit from them. And I said that because we share data with, if it's inside the US, we share data with local and state officials. As we know, insects don't know boundaries. They don't know the installation gate from the outside. So there's always a continuum of if there's an infestation somewhere, let's say mosquitoes are a problem in a particular area. Let's just say San Diego, for example, even though there's an installation there, it's going to be the same inside installation or outside installation. So we share some of our surveillance data with the local health departments, as well as what they share with us, which really helps. Thank you for that question.
Thank you, Dr. Morawo. The next question is from Taboo to Professor Dingha. She said, thank you so much for an inspiring presentation. Beyond publication and research productivity, what do you think truly distinguish a strong graduate student?
So it's a combination. I don't look at it like a single aspect that will kind of distinguish you. So it must be complementary. Research, publication, outreach, other activities that are not, just focusing on your research. So when you are looking at that productivity, is a package. You can't just say you are looking at only publications, or you are looking at only outreach. Because most often, if you are looking for a job, say you graduate and you're looking for a job, it's the entire package that counts. Most people are going to look at, okay, they're able to do good research, they're able to publish, but some other aspects of outreach, what did you do? Did you write grants? So all of those things come into that full package. So you want to be grounded on all different aspects, not just on one. I hope I've been able to answer that question.
Yeah. Yep, thank you. I think you surely did. Okay, I have another question for both panelists, and it is, what shape your approach to research, leadership, and your professional responsibility? So if you could say, what is that one thing in your life or your journey? What is that thing that shape your approach to your success?
I think I'll go ahead and say it's more of dedication. You might find yourself somewhere where you have that passion, that dedication. And I talked about persistence. And you must work hard, because it's not going to be easy. I don't know if the audience that we're talking to are mostly graduate students. So since I don't know the audience, I'm just going to talk, all around. Even when you secure a job, you have to be productive. You have to work hard. There's nothing that comes easy. So it's all about hard working, being persistent in what you do, and do it right.
All right, thank you. Dr. Morawo, you have some insights to offer in that question?
Yeah, I absolutely agree with that. Besides, resilience and hard work, I would say this for my part, I am always trying to try new things. Like I'm never scared of trying new things. And I think, that's something that I've taken from my own just build up, so far to getting here. I am not afraid to change jobs. I'm not afraid to move cross country. I'm not afraid to go outside the country. And I think that mentality really helps, especially in today's world, where you may have to go from places to places, maybe even switch your research career. Like I started as a chemical entomologist, right? I was in agricultural type of entomology, which is, most of us, and then completely switched to medical entomology, just like that, being in the military. So it wasn't a jump that I was dreading. I just knew if I had a solid foundation and I just had the drive to do it, it is possible. So, yeah.
Nice, thank you. The next question is, as we speak about role models, are there any scholars or leaders who work as influenced your career path? And if yes, how do you carry their legacy forward?
I can go ahead and just say mine. So I have a lot, but because of time, I know there are probably other questions. I'll just give a few examples. When I first started at Auburn as a graduate student, I got the chance to, and I'm glad that this happened early on in my graduate study. I went to Tuskegee University, and I visited the George Washington Carver's Museum, and my mind was just blown. And I just I looked at those things. I mean, it's very easy to look at those apparatus and all those innovations and everything and say, that they kind of look simple today, but for its time, that was a great level of just thinking outside the box. And it was from there that I just picked it. I was like, you know what? I'm gonna think outside the box. I went back, read a little bit more about him, about how, it helped people like kind of like shift the idea about research and just the little things that he put together. It was just awesome, and it just inspired my own innovative ability. Another one is Thomas Eisner. And I'm not saying this, for the chemical ecologists that I hear, they're probably like, oh, because he's the father of chemical ecology. No, not just that. It was just his attention for details, being just a pure ecologist. When I read his 2003 book called For Love of Insect, if anybody has not read that, please go read that if you have a chance. I mean, just the details at which he described insect behavior. After I read that book, it was the first year I started my graduate program. I just fell in love with insect behavior. Like that's where my passion really lies in terms of research. So those are some of the things that has shaped me. And when we're talking about like more recent people, I cannot, I must always, talk about my own advisor, Dr. Fadamro, who's also a member of IABE. It's not just because he was my major professor. He was, he went kind of above and beyond and became a lifelong mentor to me. It was just the way that he did the things and the things that he aspired for, always looking, up to that. And it's not just him, I've gone around different places, seen some of our own, mentors in the entomology line, and I think they are doing amazing work and a lot of things that I can take from it.
Nice, thank you. Dr. Dingha, do you want to add? So to repeat the question, it says, are there any scholars or leader whose work has influenced your career path? And how do you carry their legacy, if there are any?
Okay, I guess my question is very, very personal. I started at Auburn University.
Okay.
Before you. Dr. Appel was my advisor, and my project was basically lab work. I worked with the German cockroach. But somewhere the trajectory changed, and because of I'm married to an entomologist. So when I left Alabama and came to North Carolina A&T State University, I started with field work. That's my main mentor. And I have others who have encouraged me, who have supported me. but because of what I saw in what he does, it encouraged me to move on in this area. So never think that you can never change your area of specialization. You could be doing lab work today and you find something that you think you are so passionate about. Don't hesitate, go for it. Thank you.
Thank you. All right, we have some questions in the Q&A. This one is for Dr. Morawo. It's from Adeboya. It says, I am a PhD candidate with a focus on forensic entomology. I am wondering if there are any sections in the military that I can serve as a forensic entomologist. Note, I am an international student.
All right, yeah, good question. Thanks for asking that question. I would say this, if you have any background in entomology, let me just tell you what the requirements really are too. And this goes for almost all the services, whether it's Navy, whether it's the Army. They want you to have a graduate degree in entomology. Now, whether your background is medical entomology or not, that usually does not matter. Just in my case, I was doing chemical ecology and looking at agricultural pests, but I was able to make that pivot to medical entomology. So you just need a solid background. So forensic entomology, that works too. Now, as far as doing actual forensics, entomology aspect in the military, that may not be your primary thing that you will be doing if you commission as an officer, but it may become part of the thing that you'll be doing. The main focus is always going to be medical entomology. So just to get that out. Oh, as far as the starters, so the entomology is an officer only job in all of the services, and it's usually only open to U.S. citizens. So.
Yep. All right, thank you for your response. We have a comment and questions from Jennifer. She said, thank you both for your presentations and sharing your experiences. You're both active volunteers within our ESA community. Could you comment on how that has benefited you personally or professionally, and what motivates you to be active volunteers?
Active volunteers on IABE?
ESA and just overall generally.
Oh, active volunteers to ESA, right?
Yes.
I have not actually participated a lot with ESA, but I can say being a member of an association that is a major area in which my expertise is in, there are so many benefits that just reading papers that are free online and then attending meetings and interacting with people who share similar expertise that you have and learning from them is so important. And the IABE is not excluded because during most of the ESA meetings, we always have a section, and I think you saw from the presentation, several slides were shown with pictures that, as members, we come together and discuss things that are related or affecting us directly. Thank you.
Thank you. All right, we have one more question, short answer, insect related. So in what way has studying insect systems deepened your perspective on resilience? And is there a specific species or system that has mirrored or inspired your personal journey?
All right. I'm gonna take that. But before I say that real quick, just on that last question, if you are a student, my own involvement goes back to 2012, and ever since then with ESA, that was also when I started with IABE. But for ESA specifically, if you're a student, this is invaluable. You're an entomology student, you need to get involved with ESA. This is how you get your, future advisors, future jobs, and everybody. That's where everybody's looking at. Networking there I can't even quantify how much it's going to make a whole lot difference in your career because the entomology community is just that small and just tight knit. All right. With regards to the question at hand about a particular insect, I think there's so many of them, with me looking at just insect species, but generally I look at insect behavior and just how much, if you're doing research in insect behavior, a lot of times you will not get results. It will teach you how to be patient. This is something that has happened to me. I have become a lot more patient since I started doing insect behavior because sometimes they just don't want to do it and you just have to wait. So that has really taught me patience, not just something that I'm using now in research, but also just generally as a life lesson. So something that insects taught me. Yeah, I can take that on. Yeah.
Thank you. Dr. Dingha, do you want to offer any final lesson that you've learned from, insect studies and insect system that inspire your final thoughts to the audience?
Yes, so I have worked with cockroaches, I've worked with bees, but the one thing I've noticed with insects is that they tend to be smarter than we are. So whatever we're trying to do, either to manage them, they are always ahead of us. So we have to be patient and invest in our research to be able to manage this insect. And there's no specific insect that I can say is really my favorite, if that was part of the question. And here at North Carolina A&T, there are only 2 entomologists. So we are looked upon as knowing everything as far as every insect is concerned. So if somebody sees an insect, they just bring it and they expect you to know that insect. Or if there's a problem, anything related to entomology, we should know we are the experts in all of that. So right now I don't have any specific insects. I just love them.
Yeah. So thank you both to our panelists, and thank you for your final word, your final advice from your own inspiration from an.
Thank you.
Now we will move towards the closing. So if you would like to be, this webinar is organized by IABE. If you would like to be a member, we want to offer you a final IABE voice, a member's reflection. This is from Anne-Marie Robbins Baker, who is a PhD student in entomology at Florida A&M University.
I choose the word belonging, knowing that I am seen, valued, and I'm supported in a field where representation has not always been the norm. But through IABE, I am reminded that I am not alone in my journey as a scientist.
Now, if you would like to become a member of IABE, you can contact myself or any member of our executive team, Blessing Adekamoya, Tolu Agondabe, as well as Dr. Femi. Thank you for attending, and we look forward to seeing you again next year.
Thank you to our moderator, Tara Kay, and a big thank you to our panelists, Beatrice and Tolu, for sharing their academic journeys, career histories, and their passion for insect science with us. For anyone listening, you can learn more about each of our panelists and their work on our website, entsoc.org. Again, if you wanted to see the photos that our panelists shared, you can find this video recording on YouTube or our ESA Learning Center. Thanks again for tuning into Stridulation Station. If you enjoyed this episode, subscribe, leave a review, and we'll be back soon with more conversations sharing the work of our insect scientists. I hope you join us for a future conversation or even in person at an upcoming ESA meeting. Until then.